Phylogenetics. Phylogenetics. Evolutionary theory states that groups of similar organisms are descended from a common ancestor. Phylogenetic systematics or cladistics is a method of taxonomic classification based on their evolutionary history. It was developed by Willy Hennig, a German entomologist in 1950. Major reasons to use phylogenetics: understand the lineage of different species, organize principle to sort species through a taxonomy, understand how various functions evolve, understand forces and constraints on evolution, perform multiple sequence alignment, predict gene function, phylogenetic footprint. Species or gene trees. Species trees. Species tree. How are my species related? Contains only one representative from each species. When did specimen take place? All nodes indicate speciation events. Gene tree. How are my genes related? Often contains a number of genes from a single species. Nodes relate either to speciation or gene duplication events. Species tree. Species tree. Let's have a look on it closer. So this is species tree. Embryonic amniotic sac. Hind limbs, wings, underbody. Grasping forefoot. Okay. Okay. From embryonic amniotic sac, there are turtles, and then there are mammalia, and then there is hind limb swings underbody. Okay. I don't know. Okay. Then we have grasping forefoot. Then we have feathers. Then we have monon, monon, mononicus. No. Then we have no teeth apes. Then we have skull change, tri, Tyrannosaurus. Then we have here pelvis shift, and then we have enamel change. In this we have theropods, and then we have armor. In this we have stegosaurus. Okay, let's go on, move on. Let us move on. Phylogenomics, genome trees. Explore genome evolution based on large large data sets of DNA or protein sequences using entire genomes to infer a species tree. Eisen and Fraser 2003, based on maximum genetic information and average out of the anomalies, has become the standard for reconstructing reliable phylogenies. Ciccarelli et al. 2006, Dubin et al. 2006. Okay, just to remember. Okay, let's do this. There is phylogenics and tree of life. Eukarya, Archaea, bacteria. Bacteria we have Plankomycetales, Los, GC, gram-positive, thermatogeles, equifocales, spirochetes, chlamydiales, dinofocales, high GC gram-positive cyanobacteria and proteobacteria. And then we have here mitochondria and chloroplast. And then we have eukarya. Eukarya we have plantae, land plants, green algae, red algae, glaucophytes, mycetozoans, telobionts. And in Amoebozoa we have Entomoeba, diplomoniads. In Excavata we have diplomoniads, eupinoids, jacobids. In Chromel violeta, we have here cryptophytes, haptophytes, raminophiles, alveolates, radiolaria, sarcosa. This is this is it, Rhizaria. And in Petazo and Opisthoponta, we have fungi, chonicles, flagellates, and materials. And after that, we have Archaea. In Archaea, we have Thermoproteus, Crenarchaeota. We have Thermoproteus, Sulfurococales, Sulfolopales. In Eukaryota, Euchar in Archaeota, Halobacterials, Methanocercinales, thermoplast, thermoplasmatals, and archaeoglobales, methanococcales, thermococcales. And that is it.